Proteins Part 4. Biosynthesis of amino acids. In this lecture, we will study the biosynthesis of amino acid of 3-phosphoglycerate precursor family. Let us start. 3-phosphoglycerate is an important metabolic intermediate of glycolytic pathway, and it gives rise to three important amino acids: serine, glycine and cysteine. 3-phosphoglycerate is the starting material for the biosynthesis of serine, and serine then gave rise to two other amino acids, glycine and cysteine. Biosynthesis of serine. In this pathway of biosynthesis of serine, the 3-phosphoglycerate first oxidized to form 3-phosphohydroxypyruvate, and this reaction first is catalyzed by the enzyme phosphoglycerate dehydrogenase, and it utilizes one NAD+. In the next step, second, this 3-phosphohydroxypyruvate is transaminated by the enzyme phosphoserine aminotransferase, and it produces 3-phosphoserine. Glutamate is the donor of ammonia group in this transamination reaction. In the third and the last step, the 3-phosphoserine is converted to serine by the enzyme phosphoserine phosphatase, which hydrolyzes this phosphoryl group attached in the 3-phosphoserine and form the serine. As a result, the serine is produced from 3-phosphoglycerate by three-step reaction. Now the biosynthesis of glycine. Serine produced from 3-phosphoglycerate give rise to glycine. Glycine is synthesized from serine by single-step reaction. In this reaction, this beta carbon of the serine is removed by the enzyme serine hydroxymethyl transferase, and the removed beta carbon is accepted by this tetrahydrofolate, and it forms N5-N10 methylene tetrahydrofolate. As a result, the glycine 2-carbon glycine is formed from 3-carbon serine. The reaction is PLP dependent. As a result, glycine is formed from serine. There is another pathway of biosynthesis of biosynthesis of glycine, and it occurs in the vertebrate of liver. In vertebrate liver, the glycine is produced from carbon dioxide and ammonia, and in the presence of enzyme glycine synthase. It utilizes one NADH plus H plus and N5-N10 methylene tetrahydrofolate. In this reaction, along with the glycine, NAD plus and tetrahydrofolate is formed. Thus, glycine is formed from serine, as well as it is also produced from carbon dioxide and ammonia in vertebrate liver. Biosynthesis of cysteine. The biosynthesis of cysteine in plants and bacteria is different from that in mammals. Cysteine is one of the two sulfur-containing amino acids. Other sulfur-containing amino acid is methionine, and the sulfur required for its synthesis comes from sulfate, which is present in the soil. Plants and bacteria convert this sulfate into sulfide by a series of reduction reaction. And this pathway begins with when sulfate present in the soil reacts with the ATP, and it forms APS, adenosine 5-phosphosulfate. In the step second, which is wrongly written here first, this is the second step. In the second step, the One more ATP is required for the activation process, and this second step add the phosphoryl group to this third carbon, and the product form is PAPS. The product form is PAPS. PAPS means 3-phosphoadenosine 5-phosphosulfate, and this reaction second is catalyzed by the enzyme ATP sulfurylase. The PAPS thus formed is converted to PAP by enzyme PAP reductase. This enzyme releases the sulfite ion and form PAP means 3-phosphoadenosine 5-phosphate. The NADPH is the reductant of the reaction. The sulfite ion thus produced in this step third is further reduced to sulfide by the enzyme sulfide reductase. This sulfide is then transferred to O-acetylserine, and O-acetylserine then give rise to cysteine. There is another pathway of formation of cysteine from the serine, and in this pathway the serine first combines with the acetyl coenzyme A. As a result, the acetyl groups attach to this beta carbon of the serine, and this produces O-acetylserine. This reaction is catalyzed by the enzyme serine acetyl transferase. The acetyl group attached to this O-acetylserine is replaced by the thiol group, and this thiol group comes from the thiol means SH group, and this SH group comes from the sulfite reduction reaction. As a result, cysteine is produced from the serine. Now the biosynthesis of cysteine in mammals. Mammals have a different pathway to synthesize cysteine, and in this pathway methionine, which is a sulfur containing amino acid, is utilized along with the serine. The methionine provides the sulfur atom and the serine provides the carbon skeleton. In this pathway, methionine first combines with the ATP and it forms S-adenosyl methionine. The reaction first is catalyzed by the enzyme methionine adenosyl transferase. As a result of this reaction, the adenosine group gets attached to the sulfur atom. In the step second, this methyl group attached to the sulfur is replaced or removed, and it is accepted by the receptor R. As a result, S-adenosyl homocysteine gets formed from S-adenosyl methionine, and this reaction is catalyzed by the methyl transferase enzyme. In the step third, the enzyme hydrolase removes this adenosine groups which is attached to sulfur atom and forms homocysteine. Thus, homocysteine formed from S-adenosyl homocysteine by the removal of adenosine group. This homocysteine now condenses or combines with the serine, another amino acid, serine, to form cystathionine, and this reaction is catalyzed by the enzyme cystathionine beta synthase. It is a PLP dependent enzyme. In the next step, the cystathionine gamma lyase enzyme removes the amino group of homocysteine and cleaves the homocysteine into cysteine and alpha ketobutyrate. As a result of this reaction, cysteine is produced from methionine. Thus, we see that the sulfur atom of cysteine is derived from the methionine, whereas the carbon skeleton comes from the serine. As a result, the cysteine is formed from methionine and serine. Now, the summary of biosynthetic pathway for phosphoglycerate derived amino acid. Thus, we see that the 3-phosphoglycerate is an important precursor and it gives rise to 3 important amino acids, serine, glycine and cysteine. 3-phosphoglycerate first give rise to 3-phosphohydroxypyruvate and 3-phosphohydroxypyruvate then give rise to 3-phosphoserine. The serine then give rise to glycine by a single step reaction. Glycine can also be synthesized from carbon dioxide and ammonia in vertebrate liver. Serine give rise to cysteine when it combines with the acetyl coenzyme form O-acetylserine. O-acetylserine then give rise to cysteine. There is another pathway of synthesis of cysteine in the plants and bacteria. When in plants and bacteria, ATP combines with the sulfate which is absorbed from the soil. It produces adenosine 5-phosphosulfate. The 5-phosphosulfate, which is known as APS, gets converted to PAPS, means 3-phosphoadenosine 5-phosphosulfate. This PAPS now reduced to sulfite and then sulfite get reduced to sulfide ion. The sulfite get transferred and combines with O-acetylserine to produce cysteine. In mammals, the pathway for the biosynthesis of cysteine is different from that of plants and bacteria. In mammals, this methionine combines with the ATP to form S-adenosyl methionine. This S-adenosyl methionine converted to S-adenosyl homocysteine. S-adenosyl homocysteine then give rise to homocysteine, and this homocysteine when combines with the serine give rise to cystathionine. Cystathionine then give rise to cysteine. As a result, 3 important amino acids are formed. For the students of MSc Final Botany having special paper Biochemistry, I would like to suggest a book of Biochemistry by S.N. Gupta. Thank you for watching the video.